We are now to the panel discussion that will conclude Synchrony 2020. Before we dive in, I'd just like to take the opportunity to once again thank Pramila, Sarika, and the rest of the Brain Foundation for such a tremendous opportunity to bring everybody together, and really just outstanding job overall. I wish we could all be clapping. So, kudos to you guys for all your efforts. It's really been tremendous. So we've got a few questions that have been queued up. So I don't know the best way to go about really addressing them, but I'm just going to kick it off with a question and first person to speak. How about that? First question, do all of you together believe that it is time to recategorize autism as an immunological condition rather than a psychiatric one? And how can this change be brought on? As an example, MS is a very similar disease in adults. It is neither seen or categorized as a psychiatric condition, nor treated by a psychiatrist. And if it were, we would produce the similar results to autism. So, Judy, you want to start it off?
I'm looking right at me. It's funny because as you were saying that in the comment about MS, I remember when MS was first characterized, that dates me, but as an autoimmune disease, as an immune-mediated disorder, when before it was just, they had no idea what the neurodegeneration was about. So if it can happen for that, I think we can push the envelope. And I think I would like to start the discussion by saying, I don't think all of autism should be characterized the same way. I think the heterogeneity of this disorder has kind of driven us down that find the cause of autism, when just from the presentations today, you can see the heterogeneity, how important understanding that heterogeneity is, especially as we try to determine the best way to treat the kids or prevent it. And I think, so how do we make it happen? As someone who's been trying to understand the immune underpinnings of this disorder for 21 years now, I think we've made huge strides from when I first started. And I think having people like Matt, who came at like looking at immune cells in the brain from a completely different viewpoint, is really how we start to make this happen. I think, as I said, that the really good phenotyping of the kids in the context of that heterogeneity is going to be critical to getting people on board with, okay, this is this immune type of autism. I think calling something an autoimmune disease, we have such a defined word or such a strong definition for what we constitute as an autoimmune disease. That's why I like to use the broader immune-mediated disorders, because the immune system being dysregulated, it may not be autoimmune in the classical sense, and I think that kind of puts people off. So I think if we start to change how we, how we label things a little bit more would also help. And I'll stop talking so someone else can say something. Unless you want me to say something else.
Oh, hey, you got 21 years of information in your head. We've got fun. We can keep going. One thing that I'll add to this, I don't think, and I'll probably be the contrarian here, so I apologize. I don't think we're going to change the definition of autism from a behaviorally defined one. But what we will hopefully get towards, and it's weird for me to say coming out from having a biomarker company, but I think it's absolutely fundamental in that we enable a biological understanding, to your exact point, Judy, getting at better subcategorizations of autism. Maybe we can start having certain biological markers integrated into the diagnostic label, but it kind of opens up the next set of questions of, well, how do you get a harmonization agreement around that and get everyone to agree on what biomarkers are which, and who gets to integrate what, and how can we get there? So it's challenging. I mean, it really is. But I'd love to continue to keep this going around. Matthew, Alessio, Chris, Diane, anyone have anything that they'd like to add?
I do want to say something, because the same question about how do we redefine Parkinson's is challenging to field right now. It's not one disease. How do we define it as an immune condition? I started in the lab with understanding the role of neuroinflammation in Alzheimer's and Parkinson's. And I really like how Michael J. Fox says it. It's the genes that load the gun and the environment that pulls the trigger. And it's not one thing, but in drug development, 25 years in drug development, what's really frustrating is you can spend millions and bring a target forward. You never want to have a trial where you don't know that you tested the hypothesis. Whether it's a systemic peripheral target of inflammation or a central drug that's changing excitatory inhibitory balance, whatever therapeutic agent is tested in autism, we need to make sure we have biomarkers that show target engagement to have confidence that you tested the mechanism. And that's really, where I think when you asked what studies and what's the roadmap to bring together, the FDA always tells us, bring us the data to show.
Well, I have a quite biased view on the story, so I apologize to be so bluntly direct. But if you ask yourself, is there any single case of autistic, situation in which there is no a biomarker inflammation is on? And I know the answer already. No matter if you look at human beings, no matter if you look at animal models, there are always signs of inflammation. And if there is inflammation, by definition, the immune system is involved. So that this is an immune-mediated disease, I believe, is out of discussion. Again, I'm extremistic here in my statement. Question being is, as Judy was mentioning, what kind of immune-mediated, reaction you have. I believe that this is like a single starting point, branching out in so many different subgroups and regrouping again in what is the neuroinflammation that characterize, the autism spectrum. You look at animal models, so you see the microglia involved. So it means that the immune system is involved. You look at innate immune response, and sure enough, is in there. You see the adaptive immune response is in there. Maybe a subgroup is Th17 mediated, so you can define the subgroup as an autoimmune, and others is Th1 response, and therefore you have to go with a different kind of inflammatory process. But the inflammation is involved. I believe it should be a settled, discussion.
So I tend to agree with you scientifically that that an inflammatory component and an immune component is absolutely there. But getting back to clinical and diagnostic categorization of autism beyond, how do you get your psychologist, psychiatrist? How do you get those people on board to agree?
Why do you think that this is mutually exclusive? That if you agree that there is an inflammatory component, then the psychological component takes a backstage? I think that these are pieces of the same puzzle. If you look at type 1 diabetes, you have an immune response that happen to be an autoimmune response that target the beta cells that produce insulin, and you develop diabetes. Here you have an immune response that target a different organ and with different clinical outcome. I don't think that the endocrinologists are really upset at the fact that there is an immune component in diabetes. It's a fact. It's a fact. As a matter of fact, if we want to find solution, I believe this is a multidisciplinary affair. The psychology by themselves and the immunologists by themselves, the geneticists by themselves will not find the solution here. I don't think.
No, I agree with you 100%, Alessio, but I think for those of us who work with the clinicians, I've had them stand there and say, we know how to diagnose autism. We're really good at it. We've been doing it for a long time, right? So that marriage of, and I've had them say, I'm so tired of your stuff being out there and in the public, because it takes away from what we do for the families, which just astounds me, because we should all be, as you say, this should be a collaborative effort, because what is our goal? Our goal is to help the kids and the families. It's not just to further our careers. So I think that synergy and maybe the newer generation, the sort of the younger generation coming up in this field is going to be, it'll be helpful, because they're maybe a little more open to that integration, which, as I say, is key. It's working on this together, and my biggest hope is to be able to look at the genetics of the moms who have kids with autism and have the autoantibodies, and see what that or look at the genetics in the kids, see what those overlaps are. I mean, it's a little bit expensive to do, but to me, it's sort of, I'd like to have more integration than we do, as you say.
One thing that I've taken a perspective is, as a neuropathologist, I get muscle biopsies from individuals with weakness, right? There are certain unique clinical features of different muscle diseases, but I think it's somewhat analogous. The genetic forms of muscular dystrophies, might be analogous to the genetic things that directly hit the circuits, right? That are the small fraction that hit famous genes like SHANK2 or others. And that leads to the same clinical presentation behaviorally. And then, there could be polygenics that also hit those systems, but, and in fact, those may be the subset that don't show up in the metabolome or in the sort of inflammatory markers, that very small, tiny subset. What's interesting is, though, that we're getting this sort of big chunk, which has inflammatory brain tissue signatures and a metabolomic signature. And I think at some point we have to marry all that data and really use it to help us understand. One thing that, thinking across all the talks, folate receptor alpha immunity hits pretty frequently in autism. And folate deficiency would give you a systemic metabolic defect, as kind of is beginning to show, is the foundation of some of that stuff that we heard at the very last talk. So I think that, but then there are these subfractions which may be an antibody-based pure thing during, the in utero state, at least, synthesizing all these and figuring out which ones subdivide or which ones actually overlap to some extent is important. And I think we need to think of the brain as just another simple organ system, which is impacted multiple different ways, possibly, in the same common pathway that leads to the clinical presentation we have. But I do also feel that this progress is slightly obstructed by this being solely in a certain domain of psychiatry, and that, we don't have immunologists treating autism or thinking about autism. In neurology, there's MS docs, which are neurologists trained in how to treat immunologic disorders and how to think about them. But I think that that's a little bit of an artificial barrier that's formed because of the domains where this disease falls. It's been psychology, psychiatry, and developmental medicine, and they don't think about autoimmune disorders very much.
So I have a question. I don't mean, sorry, John. I just wanted to add.
No, Chris, go ahead. Yeah, please.
So I hate to be the party pooper, but at least I can play a skeptic, and then you guys can explain to me some of this. So, and perhaps here I will divulge some of my training that's closer to the psychiatric end of things than to the neurological side. But I guess what is strange to me about this is that, all the data that was presented was really very interesting and very encouraging. And it would be great news if it turned out a large bulk of this condition were actually immunologically mediated in nature, because as has been pointed out, I think there's reasons for optimism that neuroimmunology will be a pretty incredible area in the coming years. But I guess I don't understand why we're not seeing clearer signals of this at the macroscopic level, say in the neuroimaging that we've done. It's been very difficult to find some of these markers at that gross level, at least in EUAIMS, and to the extent that ABCCT can look at this there too. So is this how a neurologist would approach this differently and tell us we're just using the wrong MRI sequences, or there's something else like that happening? Someone could help me understand. It could be great news.
In polymyositis, you cannot see it radiologically.
I can give you another bigger and more, compelling example. As I'm an expert in celiac disease. This is a condition, it's an autoimmune disease, no question about that. And it's characterized by severe damage of the intestine in which the villi are gone. You do an endoscopy. So not an MRI, an endoscopy. So you look in the gut, you see nothing. Zero. Absolutely zero. It can be pale and nice and pinky and rosy and beautiful. No sign of inflammation whatsoever, unless you do a biopsy. You put the biopsy under the microscope. All this to say, looking at, sign of inflammation with imaging, it may be deceptive, because the information can be so subtle that you can't reach the level of detection by imaging, for example. This is all to say that, and again, what Judy was saying, I don't think that, clinicians should take an exceptions or a position, say, we really don't understand why you're pushing us in this direction because we got this covered. If it was the case, why we have this meeting? So obviously there is room for improvement here. This really takes the humility to sit around the table and call upon on all the experts that will eventually can contribute to moving this field forward. And I finished by saying a great lesson that I learned is there are some other conditions. You mentioned Parkinson, you mentioned MS, but diseases that are not even targeting the brain. They can teach us a great deal of what may be at play here and what will be the appropriate approach of the situation. If somebody 10 years ago, 15 years ago, would have said that cancer is immune mediated, and somebody will go to win a Nobel Prize, people will look at you like you came from Mars. So we just need to be humble and open-minded, that's all.
Thank you so much, Alessio. Just in the interest of other questions that have come up, we're gonna switch to another one that's come through. I think we could talk about all of these for hours. What would be the easiest path to get medical treatment referrals for someone diagnosed with autism, as opposed to only speech, OT, etc.? I guess kind of getting at that. So, right now, standard of care for autism is diagnostic, behavioral diagnosis, ABA, early behavioral intervention, OT, speech, etc. Beyond just, supportive behavioral treatment, what's it going to take in order to get medical treatment referrals in your mind? Alessio, you want to start?
Well, again, this is another bias of opinion I'm giving here. I believe that, compared to 10, 15 years ago, the intervention that we have now available, specifically early intervention, completely changed the landscape of the destiny of these kids diagnosed with autism. So definitely we made a major improvement of what was before. But if you allow me the parallel, we're treating the consequence and not the cause. It's like they have pneumonia that brings fever, and I take Tylenol. For sure, Tylenol will bring the fever down. But as soon as the effect of Tylenol is over, the fever will come back up unless I eradicate the reason why I have fever. So in other words, that I take, the infection out of my body. With all this intervention, we're buying time. And I don't want to minimize the importance. I'm saying yes, because we don't have an alternative, we're buying time. But the goal is really to find the target intervention. And again, we heard about the importance of genetic. We heard about the importance of the environment. of the microbiome, the metabolome, so that we can find biomarkers there. You know, there is another lesson that I dearly learn on my own mistakes and frustration of years of research. We're looking the wrong direction. And I know that I would be unpopular to say so. With a few exceptions like Fragile X and a specific mutation that I can, you know, pinpoint in, I don't know, in mitochondrial metabolism and so on and so forth, the vast majority of this genetic signature of autism, it doesn't really give me directions what exactly is going on, nor the you know, the environmental factors taking, pollution or, you know, food intolerance and so on and so forth. Each of us are genetically different, and each of us, they have a different microbiome. So there is no a normal genetics or a normal microbiome. It's the results of this interplay, i.e. the metabolic pathways, that is what we are looking for. And we are built as human beings to have a steady state metabolic pathways that will keep our blood pressure within a certain range, the glucose level a certain range, the creatinine in the urine a certain range. Anything that derails from there needs to be adjusted to go back in that steady state. If we are not capable, we develop pathologies. Same story here. If we understand, metabolically speaking, what is the derail that brings the neuroinflammation, now we have a target. So going back to biomarkers and you know, a target of intervention. And I will venture as my last, you know, statement, I don't think that anybody would disagree that it's not going to be a single intervention that will fix them all. That's the reason why patient stratification is going to be instrumental.
I sit on a committee of clinicians that take care of brain tumors. And it's very effective because in that same room, we have radiation oncologists, you know, neurologists. It's just very diverse. And I think that that is a really major limitation in autism therapeutics is we don't have the diversity of clinicians in the room treating each case. And we need immunologists as well as others. I mean, part of the problem though, honestly, is the framework. You know, you could imagine Parkinson's many years ago, they first found, well, you know, I think that there could be a variety of disorders where they're just kind of stuck in this little niche and nobody even thinks that they should participate in the assessment of that because it hasn't really been uncovered to be a relevant thing. And I think that probably there's a large audience that still exists in autism, right, that doesn't feel the immune system is quite that important. I think that Judy has run into that. I have noticed that a little bit too. It's starting to break through, but so I think that in the future, the good therapeutic programs are going to have people covering all these areas, you know, because it's not going to be a single, you know, because you will need risperidone for some of the clinical symptoms as well as somebody trying to treat the immune problems.
So, you know, I have a question for clinicians is how much of this multi-prong, multidisciplinary, you know, lack of effort on that part is because of lack of payer, you know, getting insurance payers, getting, you know, what are those codes that you can, you know, what evidence do you have so that we can, you know, get the payers to pay for that assessment?
Yeah, I agree. I mean, you know, it does require this, you know, academic environment-based research to kind of establish these lines of evidence, right? And the insurance company won't pay with, you know, I don't know how they respond to IVIG. You know, there has to be a compelling motivation for an IVIG treatment. It's interesting. I've heard some people that I've talked to where there have been people trying to treat autism immunologically, and they had to stop because their hospital, you know, the evidence wasn't there. You know, so I think that the evidence is necessary first, and it needs to be compelling clinical trials, really, honestly.
Yeah, I agree.
Real quick, lessons learned again from Alzheimer's experience. You know, there's compelling evidence as well that modulation of the immune system in translational models, even in humans, can affect outcomes of Alzheimer's. How this gets done is really important to think through, because there were studies that were planned to take drugs that were already in the portfolio to treat systemic inflammation, and then use those in a clinical trial to test how they would work in an Alzheimer's study. But for example, if you target TNF, but the study was not designed in a way where you take the subjects that only have elevated TNF. And if you take all comers, again, Alzheimer's heterogeneous, the chance that you're going to see a beneficial effect on a clinical outcome. So some lessons learned around: take a defined population with a known biomarker signature showing augmented inflammation, and then show biomarker changes showing that you actually change the inflammatory target that you're targeting, gives you tremendous confidence, at least, that you're hitting the mechanism before you go into a bigger study.
I believe that
Totally.
that doesn't nail this down exactly what is the problem. And that's the reason why a session on biomarker is so important. If you have an anti-TNF, medication that for other, indication, let's say, RA or IBD, and you do a double-blind clinical trials in autism, I am almost 100% sure that will fail. And the reason why, because you don't have a subpopulation targeted. Let's say that 20% of the inflammatory process go through TNF, and you do a clinical trial on randomized, a random group of 100 people, 80 which would not respond, that would be a failure. But if you have biomarkers say, gee, these folks, they have TNF alpha that is elevated, and now you do only on those 20%, you have 100% efficacy. So stratification is a key. And that's the reason why identification and validation of biomarkers is the first step. As Matthew said,
Yeah, it's that exact.
I'm sorry, I was saying, as Matthew said, some of the trials have been stopped because they didn't see the signal, because there was too much dilution of the denominator.
And, I will keep my company out of this because this is near to our heart, but we'll move on to another question. This has been a great conversation. I believe this will be the last question in the interest of time, but let's see. Would there be value in trying to get a rare disease designation for specific strong subtypes, genetic or otherwise? Would that incentivize research in pharma within the FDA in order to try and allow more groups that are interested in developing treatments for autism if they could get some sort of rare disease designation based on a certain subgroup? I'll open it up to whoever wants to go first.
I think this gets back to Dr. Buxbaum's effective talk in the beginning of the series, where he posed, if you had a candidate mechanism that hit a particular genetically based target, you went to a big pharma and you say, are you all in? There's whole dedicated units in big companies now for rare diseases because of the regulatory incentives and orphan designations. So, I mean, it's not the home run you need, but in Parkinson's, this is really what's fueling the 145 targets now in the pipeline. Many are genetically based, because if you just go in and take all comers into these trials that are so heterogeneous, it's really difficult to see an effect. So I say, yeah.
Yeah, I think another way it could help, just to add to that, right, is that that sort of designation could cause some kind of more organic grassroots self-assembly of the advocacy groups around that rare indication that would really help all of us help them, really. And in the absence of that, then yeah, you need to, it becomes very challenging to sort of couple the introduction of a new indication along with the therapeutic there. It can be done, and certainly it's like kind of part of the roadmap, because with the number of rare conditions, at least we think likely comprise autism, it will be required sooner or later that it happens that way. But the more that we can define in advance, and sort of, especially, if there are unique phenotypic characteristics or unique needs for those subgroups, as we might anticipate, there should be other benefits to introducing a rare disease diagnosis in advance.
Thanks, Chris. Anybody else want to chime in here?
I think that, again, as Diana was saying, defined as a rare disease will have some, administrative advantages in terms of facilitating the interest of some industrial, entities to get into development of diagnostic and therapeutics for autism. But in my humble opinion, the driving forces has to come from the base up. So in other words, really, Autism Speaks and other, nonprofit organizations really need to make a push, have to push with the legislator and say, you know what, enough. Now we have reached a point in which the definition of the disease is much more clear as before. The diagnostic can be done much earlier than before. And the understanding of the pathogenesis of the disease has been progressing enough now to we're at the point in which we can move from preclinical to clinical. But again, it takes a village. It takes a Manhattan project of people that needs to sit down around the table and say, let's do this. Because, the epidemics of autism is not slowing down. It is like any other immune-mediated disease in the Western Hemisphere, but at a much faster pace. We have the moral obligation to do something about it. It's like the COVID. So there was the moral obligation. Everybody needs to stop what we're doing and try to fight COVID. COVID kills you fast. Autism kills you slowly, morally, socially, and physically. So I think that deserve the same kind of level of attention that, this pandemic is imposing on us.
Amen. So to follow on to Alessio's, well said points, one of the questions that's come from the audience, if we had to boil that down to the top 3 strategic and tactical priorities that an autism nonprofit should undertake to accelerate the development of effective therapies, where should we focus our efforts? So Alessio, you just gave a beautiful, all-in sort of community, grassroots approach. I like the Manhattan Project for Autism. That's very well said. I want to open it up to other folks to speak on this. In your mind, what are the top 3 strategic and tactical priorities that a nonprofit should undertake to accelerate therapeutic development?
Yes.
Mention is the Brain Bank program, which historically, I think that Parkinson's became a real thing with the brain analysis. Essentially, and we know that the targets extend even beyond the substantia nigra. It's the cortical Lewy bodies. They're in the brainstem as well. We have to have that, otherwise we're completely going blind. Same thing in Alzheimer's. Those biomarkers are what we monitor using advanced imaging techniques to look at beta amyloid, look at tau. And it's really what is driving the choice of the therapeutics, what the etiology is. And if we go blind solely with behavioral phenotyping and don't get in and look at the tissue, the organ system that's broken and what's going on there, I think it's a huge mistake. And thank goodness the Simons Foundation and is really supporting this, and those are available. And honestly, the number of cases are growing. So I think that resource is going to be available. And over time, people will take the same sort of approach to it as they have in these other disorders. And they're going to develop, assays for, equivalent to beta amyloid deposits. And because they're going to be motivated to try to have a really sharp mechanistic biomarker for these diseases. But without that, I think we're really going blind.
Thanks, Matt. Who else wants to step in here?
I would say the top three priorities. Number one, to have the best of the best in each subspecialty together with industry to work together to line out a roadmap in which you highlighted where we are right now and what it takes to get to the final destination, i.e. an efficient treatment that is mechanistically, conceptualized and not, just a Band-Aid. So in other words, that will be a solution and not just a mitigation. That was number one. Number two, to expand on what Matthew said, I will definitely think that the biobank metadata of harmonized sample collection and information, clinical information collection is a must, because otherwise everybody goes independently. And even if you want to put this data together, they cannot be harmonized because you ask different questions, you develop different assays, or you collect different biospecimens. So gotta be an harmonization on data collection and biospecimens. And this cannot be done without the help of the community. There is no way. And third, put resources, both from the nonprofit organization, the government, in a single pot, and just give a final push rather than diluted these efforts in which you say, okay, we have now a consortium of 20 companies and 30 universities that are part of this, syndicate that has to put this in motion in practice, and just invest and go all in with this kind of approach rather than divided this up in different, directions that will just give you incremental science, but not the resolution of the problem.
One thing that's a little bit of a risk when I think organization is key, having large cohorts is key, having data coming in from, the behavioral realm, severity, all that sort of stuff, all the way down through the other measures. But the one risk there is that, there becomes dominant models of mechanism and thinking about the disorder. And I think that that's hurt the field a bit. It's a little bit dominated for a while. I think that the genetics has been absolutely tremendous. I think the SNPs eventually are going to really, as the numbers go up, are going to be tremendous. But I think that it's really, there's been gaps in sort of how to think about it, because it's a little bit biased towards what a geneticist knows, right? And I guess it gets back to this sort of many sort of diverse expertise, and ideally level the playing field in a way that there's not too loud of a single voice,
And so many of, can I say one thing, sir? Many of those themes that we just heard from Matthew and Alessio is exactly what we did at CPATH to start a consortium, what happened in getting AIMS off the ground. It is a lot of different groups working together, but it's this end-to-end approach, right? It's not a single target. Look what happened in Alzheimer's, right? 25 years to see where we are today. It was all in on one. But the things that I learned now that I've been at CPATH is, engaging the regulatory agencies early is so important. This is in our digital project now. We have 7 companies all equally paying. So they're not just paying to try it one at a time, so we don't duplicate effort. But that other initiatives under the FDA innovation is things like modeling informed drug development, where you can use data to develop models that will simulate trials instead of just going out and trying yet another trial, clinical trial simulation tools. And then they're big on patient-focused drug development. The voice of the patient, listening to parents. The traditional measures may not reveal what clinicians or parents think is wrong. You really need to understand the disease in a different way. So those are big picture changes that I see is coming across diseases that's making things happen a lot faster. Seems like it's slow, but it's actually, the PKD is the best example. It's happened over years to now have 10 companies in. So
great. So to kind of break this down, so we're looking for multidisciplinary consensus statements. We need standardized methodologies. We need a large amount of funds, large ends, engage the regulatory offices early and often. So what's stopping us? So we've had seven weeks of phenomenal speakers and some of the brightest minds in the world coming together on this problem. So I think the only thing stopping us is ourselves. So what can we do to start working together to move this field forward for these families?
So I think from my perspective, I think that multi-prong approach on a set of kids and parents, I mean, that's sort of kind of like an ACE type of thing, you know, a network of just, you know, everybody runs their you know, analytic process on the same kids, so that we have this sort of proof of, look, we can look at this same kid 7 different ways, and that, you know, helps us sort of phenotype. It's like the phenotyping project that, you know, David Amaral has, except I think probably a little deeper into some of the areas that we're working in as this group collectively. I don't know, what does everybody think?
Chris, you have anything you want to add?
Yeah, I guess I'm still sort of chewing over some of the themes from earlier in the conversation around maybe the diversity of the background training that's represented at the table. I know, Judy, this is part of what you were just saying, right? Is that you know, having this kind of multidisciplinary consensus around you know, not just clinical practice, but also represented, I think, in some of the papers that we're putting together. Like, I'm frankly not sure what a immunogeneticist would say about some of the gene sets that we're using to interrogate what we're seeing, and, you know, whether there may be just some missed opportunities there and on the training side to, you know, as we all with our ongoing, like, sort of hobby horses and areas of interest continue to pursue those, that we make sure that we have the right you know, diversity of trainees represented too at the table might really help. That's all I would add. I'm be very curious to hear the other panelists' ideas about how we could do this. I think there is a training angle here to consider.
I'm thinking about training. Perhaps the Brain Foundation is already doing this, but it's pretty amazing to see the young talent coming into this field. And so how do we mobilize them to be dedicated to these kind of big picture changes and how you approach diseases in a broad collaborative way?
So again, let me give another parallel that has been a great lesson for me. At the beginning, again, Celia disease was a gastrointestinal condition that was involving only kids. So the only experts working on Celia disease were pediatric gastroenterologists. We made very little progress, and I'm a pediatric gastroenterologist by training. We made very, very little progress to understand the intimate mechanism of the disease. Fast forward to 2020, because we had enjoyed the multidisciplinary approach of people, they're experts in genetics, in epidemiology, in metabolism, in microbiome, innate and adaptive immunity, and so on and so forth. Right now, there have been a total of 255 clinical trials in celiac disease for treatments alternative or complementary to a gluten-free diet that we thought that was the holy grail to solve the problem, and turned out that is not the case. It took really a multidisciplinary approach. And now we have a new breed of quote-unquote experts in celiac disease. They are not gastroenterologists. They are not immunologists. They are not experts in genetics or microbiome. They know them all. They understand everything. They take one branch and they got really deep into that, but they understand about everything else. They have a full understanding of the spectrum of what it takes, of the piece of the puzzle of the conditions. If a consortium, as John said, the only roadblock is us. Nobody would impede to go this roadmap to make this happen. If the consortium of the people will just stop, trying to solve the problem by themselves. And again, I don't want to say that, everybody is trying to defend its own, her own turf. It's just that, the life is made that way. And say, you know what, you need to pay your dues here. You need to be part of a team. You need to be, humble enough to say, I will do this, but together with others. And part of the syndicate is to train the next generation. Maybe not this generation, but the next one will solve the problem.
I'm not in the industry any longer, but when I started the autism unit at Pfizer, we had so much outreach and enthusiasm for the first couple of years, but what ended up contributing to it not continuing was a lack of perceived therapeutic targets. And I know in companies, we talked about how, well, you can look at the monogenic forms for targets, but if this meeting could come up with a perspective piece that would come out in Nature Reviews Drug Discovery, which every company reads, that would show the diversity and the plethora of potential targets based on the science, that would be an incredible win. Because I've been concerned that trying to keep companies investing with all the challenges with pediatrics and the target population and the lack of biomarkers, all the things affect companies' decisions about how to stay in. It's amazing to see Roche's dedication, but when you have a big failed trial, the leaders of the companies really think hard about it. Is this, how do we know for sure it's not going to happen again? So that's just one idea. It might be a good outcome from the meeting, considering how broad scope it was and how innovative it's showing that this field has moved along so much faster than I had thought since I worked on it years ago.
Great, thanks, Diane. Pramila, did you want to? We've got very little time left. Pramila, you had one last question. You wanted to make sure it was said.
The question I had was, as part of a public foundation with parents and patients with autism, there is a training aspect for parents as well, because the parent community is not really trial ready. They are not educated on the benefits of enrolling in clinical trials and what is expected. And that was part of our, it was an objective of this conference to do that. But as you know, if this conference was on something else like speech therapy, we would have had like 2,000 participants. It doesn't happen for medical research. So what can be done to, at the point of diagnosis, is there a way that the institutions can educate parents on joining, disease foundations or research organizations and preparing them for the concept of participating in clinical trials as it is done in other conditions? Because as you know, parents just facing the day-to-day issues is pretty difficult. And getting them to be mobilized to fundraise and to participate in clinical trials, from our point of view, that's a hurdle as well. And I think if anybody can speak to experience about how we can improve that situation, that would help us too.
I hate to take, again, the floor here, Pramila, but, I share with you my experience with Gemma. Gemma is a very, very, very demanding project. Imagine you have a child with autism, now you have a baby that is coming and will require very intense participation to a trial. And we have already 2,500 balls up in the air. What really made a difference for us to recruit? Engagement. We have an open line of communication with the families. We meet regularly on a monthly basis. They are not, the roadblock to participate is not the lack of education, it's the lack of communication. Every single time that I had the opportunity to talk with these folks, they said, you know what, I engaged my son, my daughter in a clinical trial or in a study. I never heard the results. They are using my kids as a guinea pig. I want to know the results. I want to be part of it. And the lack of communication really disengaged people, and you have drop off that, are the consequence of this. For Gemma, I I have a monthly meeting. Now with Zoom, it's even easier. And the only thing that I do, I listen and try to answer their questions and try to keep the line of communication open. And by doing that, there is also an education process why this is important. You did this for your child, but you did also for the community as a whole, so that again, you know, this generation, the next generation of kids that potentially can develop the problem will benefit from this engagement. Because one thing is for sure, if you really want to do translational science, where we were discussing whether you take the 3 things and maybe 5 and so on and so forth, you know, just talking about biobank and metadata, if the families are not involved, that's a kiss of death. That's over. That's not going to happen. And they cannot be passive in this process. They need to take ownership. It's us, scientific community, that we have to serve them and not vice versa. If you can communicate and make that happen, I believe that this will go very far.
Yeah, one thing that I'll add to this, that was great, Alessio, is I've dealt with this and certain challenges from, you know, when we were in Arkansas with Richard, we had a medical multidisciplinary clinic for autism in which we integrated clinical research, translational research, right? And the goal there, starting out, was to let families know of research opportunities at the time of diagnosis. But we ended up getting ourselves into a territorial war with the psychologists and developmental pediatricians, unfortunately. And so, well, how can we go about that? How can we address that problem? Well, being a nonprofit organization and working with academic institutions, embracing collaboration across groups and trying to get them to facilitate with you for that end goal, maybe that can sidestep some of these territorial issues that present. You know, being a nonprofit and being the voice of a patient population, you can be very, very powerful, and you guys can, you know, move barriers that, you know, people from working within may not be able to.
Just real quick, the Parkinson's Consortium I lead has 7 different nonprofit charity groups in our consortium. Very different approaches. Some do just basic research, but one of them called the PMD Alliance, is quite small, but they have used to be online, used to be in-person support networks with care partners and patients, but now obviously is in the virtual world. And it's really remarkable to see the level of engagement with them, because they host the companies to come and give talks about risk benefit of different medications and to talk about why clinical trials are important, because the companies can't go directly to patients, right? And it's a way to inform the patients and the families and the care partners, and they have special workshops for care partners that really help them stay involved and understand, you know, how important it is to be very dedicated to these kind of trials. So I don't know if I know Autism Speaks, there's so many groups that does so many things, but at least I've seen that a lot more engagement just based on direct reaching out, like Alessio said.